Hello, this is Mr. Cruz, and this is your flipped classroom lesson on scientific methods. Some of this should be a review for most of you, but we do need to go over it because it has everything to do with the science fair, which is a big part of first trimester. So let's review the steps in the scientific method. The first step in the scientific method is to identify a problem, which is always written as a question. Now, everybody has done this already for the science fair. You've identified a question that you're going to research and then experiment on. And researching is the second step. Researching allows us to understand our project a little more, understand the vocabulary, the terminology, maybe make some initial observations so that we know how to design the experiment, but also, more importantly, it will help us form our hypothesis, which is an educated guess. That's step 3. And this is what you think will happen in the experiment. It's what you think the answer is to the question based on your research. And once we write our hypothesis, we can then design an experiment to test the hypothesis. And designing the experiment is the meat and potatoes of this lesson. We're going to learn the parts of the experiment and how to design an experiment properly. Once you do the experiment, then you collect your data, you analyze it, you interpret the data, organize it into graphs and charts, and then you report your conclusion. You explain what happened. And when you explain what happened, you either accept or reject your hypothesis. That means your hypothesis is either right or wrong. Sometimes it's partially right. Whatever the case, you often repeat the experiment. Even if your hypothesis is accepted, you repeat the experiment just to see if you get the same results again. If it's rejected, you might revise your hypothesis. That means change it and then redo the experiment as well. So it's sometimes is an ongoing cycle, revising, repeating, just to be sure that you are your hypothesis is eventually confirmed and you can publish some reliable results. So we're going to pretend that we're going to do an experiment here for this lesson. And the question that we're going to work with is, how does green light affect the growth of pea plants? Let's pretend that's what my science fair project is about. And I'm curious on how green light specifically might help pea plants grow. Plants are green, maybe green light helps, I don't know. You can see a picture down here that kind of shows that. So this would be a neat experiment to test. So the first thing I would do is research, and I could use a number of things to research. Internet, I could make initial observations about plant growth, books, of course, magazine articles, other experiments that have been done that I find. I'm going to encourage you to actually interview somebody for the science fair. So I might interview maybe someone that is an expert on botany, which is plant science, to learn a little bit more about plant growth and the things that help plants grow. So let's pretend that this is our problem once again. How does green light affect the growth of pea plants? What would I need to research? Well, I need to know what light is because I'm going to be using a different color light. So what exactly is light? And I might have to research a physics book for that. I need to know what photosynthesis is. That has to do with plant growth. I've heard that word, but what exactly is it? And I need to know about how to grow pea plants properly. Do they need partial sun? Do they like a lot of water? What are the growing conditions? How fast do they grow? What should I expect? That sort of thing. What's the best season to do it, the time of year to do it, perhaps? So this is my research. So in my research about light, I would have learned that white light contains all the colors in the spectrum. You can see that here with this picture here, that you shine it through a prism and then you see the rainbow. And I also learned that things that reflect the color, things reflect the color that they appear. In other words, if I shine white light, which is all the colors mixed together, on a leaf, the only color that's reflected is green. You can see that right here. So that may help me come up with my hypothesis. I know that green things reflect green light, so I want you to stop and think for a minute. Do you think green light will help plants grow if they need light to grow, but they're reflecting green light instead of absorbing green light for photosynthesis? So I want you to think about that for a minute, because that will help us form our hypothesis. Now, our hypothesis is an educated guess. It has to be written if, then, because. If this happens, then that will happen because, and then you have some reasoning. So if this is done, then this will happen because logical reasoning. So here's mine. If pea plants are grown under green light, if this is done, then this will happen. Pea plants will not grow as well. And here comes my logical reasoning. Because the green leaves will reflect the green light and no other colors of light are absorbed for photosynthesis. So my research helped me come up with this. So whenever you write your hypothesis, it has to be if, then, because. So to review, hypothesis, educated guess. It's written if, then, because. And where we're at right now, recapping, we have our problem. How does the color of green light affect pea plant growth? I did some research. I came up with my hypothesis, and I think that green light will not be good for plant growth for peas. Now, here comes the meat and potatoes of the lesson. This is the hard part. It looks hard because you get a lot of words on here, but actually if you do it and apply it to an experiment, it's much easier. But the parts of the experiment that we need to know are the manipulated variable, responding variable, constants, which are also called controlled variables, control group, experimental group. So for the manipulated variable, for any experiment, you're going to set it up, and you're going to want to control every single part of that experiment and keep everything in the two groups. In this case, two groups, our green light group and our white light group. We're going to keep them the same, except for the one thing that we're changing, the one thing we manipulate, and that's the manipulated variable. That's the part of the experiment that has an effect on something, and it's the part that the experimenter changes on purpose. It's also called the independent variable. So that's the one variable that's different. The responding variable is the part of the experiment in which you are looking for the observed outcome on what happens. It's what you're measuring. And it's the part of the experiment that is changed because of the manipulated variable. Now here's how you can remember these. Always remember, the manipulated affects the responding. If you can remember this, you do MV, arrow, RV. And then you can apply that to the question. So how does green light affect the growth of pea plants? How does green light, is my manipulated variable, or the color of light, affect, that's my arrow, the growth of plants? The constants are the parts of the experiment that remain the same. They're your controlled variables. They're everything in all the test groups should be the same conditions except for that one manipulated variable. And so these are called constants. Our control group is the test group that is the normal standard conditions so that you can compare your results. Some experiments don't have a control group, but many do. It's important to have a control group because I would not know how good green light works unless I compared it to plants grown in the sun or under white light, which is their normal growing conditions. I mean, they might grow, green light might grow, but how much does the green light plants grow compared to the normal conditions? And our experimental group is the group that we're changing. It's the group with the manipulated variable in it. So a little bit more about the control group. The control group is the part of the experiment that you set up so that you can have a good comparison in your experiment. So this is a comic I found, and it shows these mice that are being tested with some sort of experimental test drug or something. And so these mice are looking over at these mice, and these mice are going crazy. They're like, ah! These mice were the test group. They were the experimental group. They were given some sort of crazy drug or something to see if it worked. And these mice were the control group. And they're saying down here at the bottom, well, I guess we're the control group because nothing's happening to them. They're the norm. They were not given any test drug or anything. And these mice in the experiment were given the test drug. And you can see it's kind of making them go crazy. And so this kind of makes a little fun of the whole control group concept. So, like if I was growing plants with fertilizer to test the fertilizer, I'd also want to grow plants without any fertilizer as my control group because that's the normal standard conditions, and I wouldn't know how good the fertilizer worked unless I compared it to plants without fertilizer. So here's our very, very simple experimental setup for our green light pea plant tests. And so you can see I have everything listed here that I want you to try to answer. And I have a general setup showing what's going on with the experiment. I have the plants grown under green light, and I have pea plants grown under white light. There's seeds in here, and we're going to water them the same each day. We're going to have the lights on the same amount of time each day, the same amount of topsoil, same size pot. The only thing that's different is the green light group and the white light group. So I want you to answer these questions, identify what the manipulated variable is, what the responding variable is, the constants, the control group, experimental group. Tell me what type of data you would collect and how you'd collect it. Is it quantitative or qualitative, and how would you collect data? And how could this experiment be improved? So I want you to pause the video on a sheet of paper, write down these answers, and that's what you're going to turn in for points today. So pause the video and then try this. Here are the answers, just so you can go over your answers and understand it. The manipulated variable was the green light. That's what was changed on purpose, and the only thing that was changed on purpose by the scientists performing this experiment. And our responding variable was the growth of the plants. And remember, I said the MV affects the RV. So here, our green light was affecting the growth of plants. The constants are controlled variables, where the wattage of the bulbs would have to be the same. The type of soil was the same. The size of the pot was the same. The time under the light was the same. The amount of water was the same. All these things would have to be the same so that the scientists knew that if there were any differences in the growth, it was because of the green light. Our control group is the white light group. You couldn't have done this experiment just with the green light on its own because you wouldn't really know how well green light worked until you can compare it to the normal conditions, white light. So the experimental group was our green light group. We would have quantitative data. We'd measure the height of the plant. You can maybe measure the mass of the plant. Now you could also have qualitative data. You could say, well, the plant looked greener, the plant looked healthier, the plant was wilted or wasn't wilted. Those are qualitative observations, but we also want some good numbers. Quantitative data is better. How could I improve this? I would have more trials, more plants for sure. You couldn't do this with just one plant in each pot. That's not enough test subjects. You need a lot of test subjects in any experiment or a lot of trials. You also, just out of curiosity, might test more colors of light other than green to see if other colors worked as well. So this is a quick lesson on the scientific method and mostly on parts of the experiment. As this video finishes out, it's going to flip through other slides that we're going to eventually probably go over in class at some point. But see me if you have any questions about this, and I will see you in class. Bye-bye.